Good day, everyone. So today we'll be looking at biochemical reasoning. So over the years of teaching biochemistry, I realized that most students are interested in cramming the pathways, writing the exams, passing the exam without actually getting the core concepts of biochemistry. So in this lecture series, I'll be looking at different aspects of biochemistry and the reasoning, how we can actually apply them in our day-to-day lives. So the first, we're going to look at different questions with respect to what is happening around us or how our body respond to environmental changes or physiological changes, or maybe in disease condition, how our body respond to disease condition and how best we can manage happenings around us that affect our biological system. So let's take the first question. Let's take the first question. Nevertheless, my name is Raymond Ibe. So if you have not subscribed to this channel, please do well to subscribe and click the notification button for more series of this lecture. Now let's look at this question. See, after intense exercise, lactate levels in the blood increases or your blood increases, but then decrease over time. Explain this phenomenon and why it is crucial for recovery. Now, what we look at first is what happens during exercise. In exercise, the the glucose that provides energy in exercise is converted to lactate via a series of enzymatic reaction. Usually, we look at the glycolysis, maybe where we convert glucose to glucose 6 phosphate, glucose fructose 6 phosphate, fructose 1 6 bisphosphate, aldolase enzyme, and the rest. But I'm not going to bore you with the whole sequence of glycolysis. But we know that the end product of glycolysis is pyruvate. Now, the pyruvate is decarboxylated by pyruvate dehydrogenase complex that goes into to produce acetyl-CoA that goes into TCA cycle for production of NADH, FADH2, and some molecules of ATP. But in exercise, because of the active exercising muscle, the oxygen supply is no longer sufficient for glycolysis to keep running on aerobic glycolysis. So the cell will have to tilt towards anaerobic glycolysis where lactate level or lactate is produced. So in anaerobic glycolysis for exercising muscle, because of the inadequate supply of oxygen, because without oxygen, TCA cycle will not take place, electron transport chain will not also be functioning. So now for those muscles that are actively exercising, instead of producing acetyl-CoA, pyruvate is converted to lactate via the enzyme called lactate dehydrogenase. This enzyme converts lactate and pyruvate to lactate with the generation of NAD plus, because that particular step requires NADH, which is produced in glycolysis. If you remember the first step in the oxidative stage of or let's say the first oxidative stage step in glycolysis, which is the conversion of glyceride 3 phosphate to 1,3-bisphosphoglycerate. So that particular reaction step, which is catalyzed by glyceride 3 phosphate dehydrogenase, requires NAD plus to run. So, and when that NAD plus comes into that particular step, NADH is released. The NADH is released, you have to, for more supply of ATP. But in anaerobic case, that NADH that is produced in that step will have to be used by lactate dehydrogenase to convert pyruvate to lactate. And once that is done, there's a regeneration of the NAD plus that enters, that goes back to glycolysis and keep glycolysis running. So let's look at the little answer I actually put up here. Now during exercise, the high glycolytic rate due to limited oxygen leads to lactate production from pyruvate via fermentation. Fermentation is in the absence of oxygen usually. Now when exercise stops, oxygen becomes available again, allowing muscles to convert lactate back to pyruvate through gluconeogenesis. Gluconeogenesis is production of glucose from non-carbohydrate precursors. So you can see here that the pyruvate, the lactate that is being produced as a result of anaerobic glycolysis, we have to be converted back to pyruvate because now oxygen is now available for the cell. So there is a need for reconversion of the lactate back to pyruvate, then decarboxylation of the pyruvate by the pyruvate dehydrogenase complex, and subsequent entry of acetyl CoA into the TCA cycle. So this pyruvate can then be used for aerobic respiration and ATP production, replenishing depleted energy stores. So this is called the lactate shuttle. This process is essential for recovering from exercise and ensuring efficient energy utilization. That is when we talk about the energy currency of cells, we talk about even with the vast array of biochemical reactions occurring in a single cell, the cell is still perfect in maintaining what we call the economy of the cell. So let's look at another question here. So you have two proteins with the same amino acid sequence, but different three-dimensional structures. Explain how this is possible. I believe that in our primary protein chemistry, we have been taught that the sequence of the amino acid is the most determinant factor in the structure or the three-dimensional structure of the protein. But we also know that there are other factors that promote the stability of that protein. Example is the amino acid side chains. The amino acid residue side chains, because of their interaction, they can actually cause stability of that protein. We can also look at the temperature. We can also look at pH. These are different factors that can make two proteins with the same amino acid sequence have the different three-dimensional structure. So we have the hydrostatic attraction, we have the electrostatic attraction, we have the hydrophobic interaction, we have the hydrophilic interaction, we have the van der Waals force. So these are other conditions or other technique. Let me not put it technique. Let me just say these are other factors that contribute to the stability of a protein. So it's not just the amino acid sequence of that protein. So I believe we should take note of that. So protein folding is not solely determined by amino acid sequence, but also by interaction between amino acid side chains. So for example, you have amino acids. One is let's say aspartate, one is lysine. And lysine is actually a positively charged amino acid, while glutamate is a negatively charged amino acid. So because of the difference in ionic charge, there can be interaction between both of them, and that will contribute to the different stable conformation of the protein sequence. So let's look at another question. So this lecture series will be looking at different questions that have question that might encounter throughout the course of studying biochemistry. And please don't forget to share this with your friends, share this with your colleagues in biochemistry, and do not forget to click the subscription button. So look at this question. You have two enzymes with same amino acid sequence. One enzyme is active at high temperatures, while the other denatures and loses activity. Explain why this might happen. This same question applies to the previous question. I was teaching my student this question yesterday. After giving them the first explanation, I asked them to give an explanation for this. They couldn't give me the explanation. So I realized that the fundamental problem is on the understanding of biochemistry. So you can see that here we are using the word enzyme. But in the previous one, we used the word proteins. So you have two proteins with the same amino acid sequence. Here we have two enzymes with the same amino acid sequence, which is the same thing. Totally the same. So one enzyme is active at high temperature, while the other is denatures or loses activity. Explain why this might happen. The same answer in the first one is also applicable here. So yeah, look at it. So protein stability is influenced by multiple factors beyond sequence, which we've explained in the earlier question. Now the temperature, the high temperature enzyme likely has a stronger intermolecular bond, which is the hydrogen bonding, the disulfide bond or disulfide bridge, which is contributed by the cysteine residues, and interaction between amino acid side chains that maintain its folded structure and active sites. So during heat stress. So the other enzyme may lack these stabilizing features, leading to unfolding and loss of function at high temperatures. So you can see that both answers are applicable. Just say we use enzyme, and the other case we use proteins. So look at this. We have a protein is translated from mRNA but does not have its full function until phosphorylated by a kinase. How does this modification affect the protein structure and function? For example, we look at this is a very sweet question. This is a very sweet one. So during translation, what is translation? Translating the code in the mRNA to protein sequence. That is translation. Now, after the protein must have been translated, now we say but does not have its full function until phosphorylated. So what does phosphorylation does to the protein? Phosphorylation is addition of a phosphate group to the protein. When we talk about kinase, we talk about enzyme that phosphorylate. Usually, the phosphate group comes from ATP. So the kinase will phosphorylate the protein, and that phosphate that is added to the protein will create a negative charge environment. Of course, you know that phosphate is maybe PO4, PO4 3 minus, PO4 2 minus, depending on the phosphate group. So now this actually contributes to the negative charge of that protein. And there are some amino acid within that vicinity where the amino acid might be positively charged. Because of the positive charge amino acid in that vicinity, it will react or it will form an interaction within the phosphate group, leading to more folding or active more folding of the protein, which will result to the activity or full functionality of that protein. And which part of the amino acid is usually phosphorylated by the kinases? Usually there are those amino acids that have a hydroxyl group as their functional group in their arrow side chain. An example is threonine. So we have the kinases that phosphorylate threonine. We have the serine, and we have the tyrosine. We have the tyrosine kinases, serine kinases, and threonine kinases. So these are examples of kinases that phosphorylate either the threonine residue, tyrosine residue, or let's say the serine residue. So upon phosphorylation, there is negatively charged phosphate group, which will contribute to the ionic charge of that protein, and that ionic charge of that protein can make the protein to fold into the desired conformation that can enable the protein to carry out its biological function. So we'll end this video here. So do not forget to subscribe. Do not forget to subscribe and click on the notification button for more videos on biochemical reasoning. So let's take a look at the question. Phosphorylation adds a negatively charged phosphate group to a specific amino acid side chain. This can alter the protein's overall charge distribution, potentially influencing its interaction with other molecules, its shape, and the ultimate its activity. For example, phosphorylation might create a binding site for another protein, activate an enzyme, or trigger conformational changes that alter the protein function. So that is the end for this lecture. Feel free to subscribe and click on the notification button. Do not forget to throw in your questions and click on the like button. Thank you.